right, so here we are, ready to move on to oxidative phosphorylation in our biosynthesis of ATP part 2. So here we are thinking about these paired chemical reactions called oxidation and reduction. Oxidation is where electrons are taken away from a molecule, and then they have to go somewhere. You've got these electrons now that have been kind of yanked off their previous molecule, and you have to put them onto another molecule. So when you add them to the other molecule, that's called reduction. So it's essentially just taking electrons from one thing and giving them to the other. They're usually referred to together instead of oxidation reduction, just as redox is sort of a shortcut. Now, these redox reactions are going to be a key part of the electron transport chain. That's really what the electron transport chain is. It's oxidizing one thing in order to reduce another thing. We're just bouncing electrons down a series of proteins. The final electron acceptor is oxygen, so that's why it's aerobic. You require oxygen in order to do the electron transport chain. Okay, so without oxygen present, then your electron transport chain does you no good. So really all we're going to be doing is taking electrons from things that we made in the Krebs cycle, specifically NADH and FADH2, and we'll be oxidizing these and we're going to be reducing parts of the electron transport chain, which is just abbreviated as ETC, the electron transport chain. Now all of this is happening in the matrix of the mitochondria, like right up on the inner cell membrane, on the inside of that inner cell membrane. And it's going to make a lot of ATP. Now, if you think back to the slide that came right before this, so the very end of our last video, we had our symporter and our antiporter. That one was bringing phosphate into the matrix. Another was bringing ADP into the matrix while removing ATP from the matrix. So we have those two carriers on this picture also, where here they called it by a slightly different name. They're calling it the phosphate hydrogen symporter. On our previous slide we called it the phosphate translocase. So that was this right here. This one over here, this is the antiporter here, where we have ADP going in to the matrix, and we have ATP that was made by the electron transport chain going out. Here they're calling it just the ATP ADP translocase. In our previous slide, it called it the adenosine nucleotide translocase. So it would be this one here. So same two carriers, these two, just somebody calling them a different name. So I just want you to get used to things sometimes having different names and just being able to recognize when that happens. Okay, so remember, this is just the source of phosphate so that we can make ATP. This is a source of ADP so that we can make ATP, combining it with the phosphate. And then once we've made the ATP, we got to get it out of there so that the cell can use that ATP. Now, how do we make the ATP? We know that the electron transport chain is part of it, but there's a mechanism by making the ATP. It's not just like a magical, like, okay, boom, electrons are bouncing all over the place and yay, we have energy. There is a protein called an ATP synthase, which was also on the last slide, but I didn't talk about it. But there it is. Here's the ATP synthase. This makes ATP as a result of the electron transport chain. So we're going to see how the electron transport chain drives this particular molecule to make ATP. Now, just notice before we get into the details of that we have in this protein hydrogen moving from the intermembrane space into the matrix. So the hydrogen is moving through that. And then almost like magic, we have ADP turning into ATP. It's not magic. It's something called chemiosmotic phosphorylation. And I think the easiest way to explain that is to liken it to a hydroelectric dam, like the Conowingo Dam. You know, it's that big dam on the Susquehanna River up between, where is it? It's right between Harford County and Cecil County. It's on Route 1. If you ever go up that way, it's kind of not a lot up there except for this dam and a nice little park with eagles and stuff. So it's actually a really nice place to go. Kind of far though, but anyway, I'm digressing. That dam takes water moving through the dam and it creates energy out of it that can be used as electricity. So in the same way that a dam makes water, moving water into electricity, and then the water can continue on its merry way down the rest of the Susquehanna River into the Chesapeake Bay, this is the same process, essentially, that this protein is going to just harness that kinetic energy of the moving hydrogen ions as they move through. And it uses that kinetic energy to add this phosphate that came in to the ADP that also came in to create ATP. So chemiosmotic phosphorylation is using the kinetic energy of moving hydrogen to add phosphate to ADP, and that gives you ATP, and that ATP can then move out in exchange for more ADP. So just like a electric dam. Okay, so if everybody's with me, we will move on. Okay, so here's our Krebs cycle again. It's not our first time seeing the Krebs cycle. This is the same exact diagram that we saw before. So we have our pyruvate. We convert it into acetyl-CoA thanks to pyruvate dehydrogenase. That's that enzyme that converts pyruvate into acetyl-CoA. Okay, and then we enter into the Krebs cycle, and we go around and around and around, and we create a little bit of energy, and we create our NADH. Where's our NADH? There's some NADH, there's some NADH, and some FADH2. Those are able to go into the electron transport chain. So these two are what we're looking for. We want to oxidize them and use the electrons that we've taken from them in order to bounce those electrons down the electron transport chain, reducing each protein in the electron transport chain and then oxidizing it in turn until we reach oxygen. Now, just as a little aside, I just want you to remember that we can also take fats and proteins and we can convert them into acetyl-CoA, just like we did with our glucose here, our carbs, our carbohydrates. So we can take our glucose, turn it into pyruvate, make acetyl-CoA. We can break our proteins into amino acids, and a bunch of other stuff is going to happen before we get acetyl-CoA, but we can still get acetyl-CoA out of it. Same thing with the fats. We can break those into their component fatty acids, breaking them down. A lot of other stuff again happening, but we can eventually get acetyl-CoA out of that. And then the Krebs cycle is the same. It doesn't matter what was our initial substrate. Was it a carb? Was it fat? Was it protein? it doesn't matter if we have acetyl-CoA and we have oxygen, we go into the Krebs cycle, the Krebs cycle will be the same. We're going to get NADH and FADH2 for our electron transport chain. Okay, so here is our electron transport chain. These are the specific components of it. We're going to see a couple other diagrams that show this in maybe more of a cartoonish kind of way, but here it is just sort of listed out so we can get the gist of it. Essentially, just remember, we're looking at NADH and FADH2. And we just want to take an electron and we want to bounce the electron from those molecules along the electron transport chain until we finally reach the end of the electron transport chain. And then we can do our chemiosmotic phosphorylation through our what's that? ATP synthase at the very end of the electron transport chain. So we're just doing redox reactions here, bouncing that electron from one molecule to another. So here we have NADH. We take the electron off and we bounce it onto the electron transport chain. That leaves us with NAD+. That's really super not clear on the screen here, is it? So this is NADH. And we take the electron away and we have NAD+. This is accomplished by NADHQ reductase is what this says. We know that's an enzyme because it ends in the letters ASE. Okay, now here we have a protein. This is called ubiquinone. So that electron is getting added there. Okay. Meanwhile, FADH2 is also having its electron taken off so that we get FADH+. And that's being accomplished by this enzyme called succinate Q reductase. Again, we know that's an enzyme because of the ASE. Okay. And then we're going to go on to this next protein, which is cytochrome B. C1. So there's our cytochrome protein. Then we go to another cytochrome protein, that's cytochrome C. And then here's this enzyme, cytochrome C oxidase. It's going to oxidize cytochrome C. And we're going to add that to our final electron acceptor. And we end up with a little bit of water as an aside because of hydrogens. I'm kind of ignoring the hydrogens here. I'm not super worried about the hydrogens. So essentially what has happened, so just as an overview here, is that the electrons have been taken from NADH. We'll just follow NADH first. So take the electron away, using an enzyme to start the whole oxidation process. We're oxidizing NADH. We're using it to reduce this ubiquinone, which is in the electron transport chain. So now ubiquinone is reduced. We then oxidize ubiquinone, use that to reduce cytochrome BC1, and which is a protein, part of the electron transport chain. So that is now reduced. Then we oxidize that, use it to reduce cytochrome C. So now cytochrome C is reduced. Then we oxidize cytochrome C using this enzyme here, cytochrome C oxidase, and we are then going to have our final electron acceptor reducing oxygen. Meanwhile, all those electrons are just, think of them bouncing along. In the same situation with FADH2. Electrons being taken, it's going to enter into the electron transport chain and bounce all the way along until we get to oxygen. Now, meanwhile, we have hydrogens just being sort of sent out in every direction as we are doing this whole process. And that hydrogen is what's going to be moving through that ATP synthase. Okay. Okay, so let's look at the picture of the electron transport chain, a little bit easier to look at as we can see just the various parts of it. So the NADH dehydrogenase complex. So that would be what we had here. So another fun situation where we just name one thing different names. So the same kind of idea. So this NADHQ reductase is part of the NADH dehydrogenase complex. So just to make it more complex than it needs to be. But this is the enzyme that is part of that NADH dehydrogenase complex. So NADH is sending its electron, or it's having its electron pulled off of it in this complex, or as part of this complex. That electron then, we'll just follow the electron to the red color. The electron goes to ubiquinone, then it goes to the cytochrome BC1 complex, then it goes to cytochrome C, so there's the electron still. Then the cytochrome oxidase complex, here we go again, naming it something different. So cytochrome C oxidase in the cytochrome C oxidase complex. Okay. So this enzyme, cytochrome C oxidase, is part of the cytochrome C oxidase complex, just like the NADHQ reductase is an enzyme that's part of the NADH dehydrogenase complex. Okay, so there goes our electron through the cytochrome oxidase complex and then on to oxygen. So there goes our electron. Now hydrogens are, see how this is all on the inner membrane here, so this is the intermembrane space. Oops. Out so you can see. So in here would be the mitochondria. Sorry, my video tried to stop itself again. Luckily it didn't. So this is the mitochondrial matrix in here. Okay, so the hydrogens are ending up out here. And then they're going to move through that ATP synthase. We have ATP in here because we had ADP plus phosphate. And the ATP can just be exchanged for those hydrogens. Okay, so just like the dam, we're creating that energy, that kinetic energy, to add those phosphates to the ADP and then to be able to get the ATP out of there. Okay, so we take the electrons, we bounce them down, the electron transport chain, finally accepted by oxygen. Meanwhile, those hydrogens are being pushed out into that intermembrane space where they can then be passed through the ATP synthase molecule, which provides the energy to make the ATP and transport it out into the intermembrane space and then finally out towards the cytoplasm where the cell can use the actual energy that you just made. Okay? Okay, now the oxidation of one NADH molecule is going to be able to make you more than one ATP. So several ATP molecules can be made just by NADH. Just this one NADH. And remember, you're getting multiple NADHs as the Krebs cycle turns and turns and turns. You're getting more and more NADH. So you can get a lot of energy just from one. So imagine how many you can get from multiple NADH. Okay. And here this is saying that NADH is the electron donor for the electron transport chain. So is FADH2. That also came from the Krebs cycle. Okay, but it's going to enter into the electron transport chain at a slightly different spot. So here is our electron transport chain again. Okay, and again, it's a different image, so it's using slightly different terminology, but we can liken. We'll just use both of these images here. I'll just kind of flip back and forth. So we're just using complex 1, complex 2. These are all part of the NADH dehydrogenase. Oops. Spelling is helpful. Let's do it on the white part so we can see this better. And the FADH2 is going into complex 2. So where they have complex 1 and complex 2, these are both part of where we enter the Krebs cycle. So NADH is going into complex 1, FADH is going into complex 2. Both are oxidizing something. The NADH is being oxidized by complex 1. FADH is being oxidized by complex 2. Okay? So on this picture, this would be complex 1. We don't have complex 2. Let's kind of put complex 2 on our picture here. And that is for FADH2 to go into. Right there. Okay. But here is ubiquinone. So that's the same right there. Okay. So let's just start from the start here. NADH, that's an NADH dehydrogenase complex. It oxidizes NADH, so we take the electrons off of NADH, we end up with NAD+. The electron is going to be bounced to ubiquinone. The hydrogen goes into the intermembrane space. Meanwhile, FADH2 is going to be oxidized by complex 2. So complex 2 is taking the electron off of FADH2, just like complex 1 did to NADH. That electron is going also to ubiquinone. Okay? So the electrons from NADH from complex 1, then the electrons from FADH2 from complex 2 are going to be used to reduce ubiquinone. Now another name for ubiquinone is coenzyme Q, so that's just another name for the same molecule. Now ubiquinone, we will oxidize that and use it to reduce complex 3. So here we are likening this to this one. So our cytochrome BC complex okay. So that is accepting the electrons. It's taking the electrons from ubiquinone, more hydrogens going into the intermembrane space, and the electrons, meanwhile, are being used to reduce cytochrome C. Now, once cytochrome C has been reduced, then it will be oxidized. And here in complex 4, so this is just where we have this situation, cytochrome oxidase complex. This is what is going to give the electrons to the oxygen. Final electron acceptor, even more hydrogen put out into the intermembrane space, which can then move through the ATP synthase, and ATP can move out. Okay. So I don't want you to feel super confused because of all these different terminologies on the pictures. The reason why I use the pictures with a different terminology is so that you can get used to looking at something, interpreting that, okay, this is a familiar thing. It's being called something slightly different, or it's being referred to in a slightly different way, but I recognize the thing that it is and what it's doing. So this is the electron transport chain. This is the electron transport chain with FADH added into it. Here they're just doing the NADH. They're not worrying about the FADH at all, just doing NADH. But it shows the ATP synthase pretty nicely here. Here, also the picture is upside down, so the intermembrane space is down here. Oops. So not upside down, it's just a different orientation where we have the matrix here. So just flipped from the other way. So I just want you to be able to see these things, recognize these things as things that you are familiar with. You have seen them. And like, okay, yeah, now they're calling it complex 1, the other one called it all this. And remember, you don't have to memorize these things. You just have to be able to interpret these charts, these pictures that are labeled, and you need to be able to understand what's going on with them. So really, in a nutshell, I know you're probably like, stop talking about the electron transport chain already, but I just want to go over it really fast one more time. Let's just think of it big point of view without all the complex names involved. But we're taking NADH and we're taking FADH too. We need to oxidize them. We want to take their electrons away. So we have these two places that can do that. Complex 1, this one, is for NADH, oxidizes NADH. Complex 2 is for FADH2, so it oxidizes FADH2. Both of those things then are going to take those electrons that they just stole from NADH and FADH2, and they're going to use it to reduce ubiquinone, the coenzyme Q. Then we oxidize ubiquinone, where complex 3 is now taking those electrons away from ubiquinone and bouncing them along to cytochrome C. Meanwhile, hydrogens are getting pumped into the intermembrane space, which is going to be important for our chemiosmotic phosphorylation. Cytochrome C gets oxidized, so we're taking the electrons away from that, using it to reduce the oxygen, which is the final electron acceptor, and that's happening in complex 4. More hydrogens getting pumped out, a little bit of water being made as a byproduct, but those hydrogens are going into the intermembrane space, so the hydrogens can move into the matrix. That kinetic energy is enough to create the ATP, and we can then pass that ATP back across the membrane into the intermembrane space and then out towards the cytoplasm. So there, that is the worst of the electron transport chain. Now, everything I just said is written here as a list. I'm not going to read it to you again. I feel like I've beaten this electron transport chain to the fullest extent, but we can just use this to study just its the bullet points of what's happening in the electron transport chain with a picture of just the ATP synthase and the chemiosmotic phosphorylation happening there. So just make sure you read that, use it as part of your studying. Okay, now our last slide for this video is poison. So this is talking about things that prevent redox reactions from happening, or things that stop the hydrogen from being able to go through the ATP synthase. So things that can block the redox reactions or keep the electrons from bouncing from one place to the other are poisons like cyanide and carbon monoxide. Carbon monoxide comes from burning fossil fuels, so if you're like stuck in a small room with a running bus, you don't want to be in that small room, or you don't want to sit in a garage with your car running. You don't want to do stuff like that, because carbon monoxide is something that is toxic, and it can help or not help. It can stop the flow of electrons. Cyanide, that one is a little bit harder to come by, which is good. But fires, so like a house fire or something like that, or a big building fire, can release cyanide. There's certain foods that have cyanide in them, or that are turned into cyanide by the body, like apricot kernels. I can't imagine anybody sitting there eating a bunch of apricot kernels anyway. Like the pits inside of an apricot, they're pretty hard and gross, but you don't want to eat them because of the cyanide. It'll mess up your electron transport chain. But I found this picture. They used to use all kinds of weird stuff as medication back in the 1800s, 1700s, even the early 1900s. They were still using cyanide, I think. So this was actually, used as like a medicine at some point, which is, not a good thing to do. Now, there's also, if we don't let the hydrogen flow through that ATP synthase, or if we let it leak across the membrane instead of being constrained to just the ATP synthase, then that is bad, because you need that hydrogen to go through the ATP synthase, or else you're not like getting the kinetic energy, you're not harnessing it. So it would be similar to like letting the Susquehanna River roll around through, not the dam, but to go around it in another direction. Then you don't get the electric energy. So there is a protein that can happen, or that can, sort of a protein that is a natural protein to have on the membrane, but that if you have too much of it, it would cause a problem. It's called an uncoupling protein. It's essentially just a leak channel for hydrogen. So if you had a condition that caused you to overexpress that uncoupling protein, that would essentially allow you to bypass the ATP synthase, which obviously is not good. You wouldn't be able to make energy. Okay, so that does it for the electron transport chain. So we're all done with that.